Hello, my name is Charles from the GIA lab, and I am presenting the role of autophagy in controlling expression and secretion of insulin peptides. Insulin growth factor regulates development and lifespan in both mammals and C. elegans. It is also closely related to metabolism and autophagy, a cellular cleansing and recycling pathway. When IGF signaling is reduced in C. elegans, both lifespan and autophagy are increased. Our lab has established that intestinal autophagy regulates expression levels of 6 insulin-like peptides, including INS1, in IGF signaling-reduced worms. DAF2 is a receptor for IGF. DAF2 mutant has a temperature-sensitive mutation that increases lifespan at 20 degrees Celsius. ATG18 is an autophagy factor, and mutants have decreased lifespans. Real-time PCR shows that INS1 is an excellent candidate for investigation first. Chart A shows that of these insulin-like peptides, INS1 mRNA levels increase threefold from DAF2 mutant to DAF2 ATG18 double mutant. Chart B shows that INS1 is the only peptide to also return nearly to DAF2 levels when there is an intestinal recovery of ATG18. This demonstrates that ATG18 works cell non-autonomously to regulate INS1 within the brain-gut axis. However, the specific neuron type that expresses INS1 is unknown. To fill this gap, our working hypothesis is that INS1 will express a certain level in chemosensory ASI neurons in wild-type worms and increase in IGF receptor DAF2 mutants. This hypothesis was formulated by a previous paper and our lab findings. Specifically, there was an upregulation of INS1 in DAF2 mutants shown by Hung et al. Our lab has established that autophagy in the ASI neuron plays an essential role in DAF2 Dower recovery. To begin this process, we generated a tissue-specific plasmid. It consists of isolated INS1 and ASI neuron promoter PGPA4. We also tagged it with green fluorescence protein, GFP, to later visualize under a specialized microscope. We then microinjected the plasmid into the gonadal arm of young adult wild-type and DAF2 worms and grew them at 20 degrees Celsius. GFP expression was visualized under an axial imager 2 microscope. This slide shows INS1 GFP expression in the ASI neuron in wild-type and DAF2 mutants. Statistical analysis indicated that about 30% wild-type worms are GFP positive. However, in DAF2 mutants, about 55% are GFP positive. Therefore, INS1 GFP expression is increased in DAF2 mutants. Our future plans include the insulin peptides INS1 and DAF28. We will be comparing their protein secretion in wild-type, DAF2, ATG18, double mutant DAF2 ATG18, and the double mutant with intestinal rescue of ATG18. This will provide greater insight into the role of these insulin-like peptides in autophagy and lifespan. I would like to give special thanks to my PI, Dr. Gia, and PhD candidate, Ms. Nicholas.